Hello friends, welcome to BMH Learning. In this video, we will discuss about single-strand conformation polymorphism. Single-stranded conformation polymorphism is a screening method that allows you to identify different genomic variants in a large number of samples and in a broad range of organisms from microorganisms to humans. Now the question is how the genomic variations are detected. So SSCP analysis detects sequence variations or importantly mutations through electrophoretic mobility differences. What does it imply? So basically, the principle is that the DNA samples are subjected to gel electrophoresis in known denaturing conditions. The electrophoretic mobility of double-stranded DNA in gel is dependent on strand size and length, but is relatively independent of the particular nucleotide sequence. However, the electrophoretic mobility of single strands is noticeably affected by very small changes in sequence, possibly one changed nucleotides out of several hundreds. But the question still arises, why these very small nucleotide changes are detectable in single-stranded DNA? It's because of the relatively unstable nature of single-stranded DNA. So, basically in the absence of a complementary strand, the single strand may experience intra-strand base pairing, resulting in loops and folds that give single strand a unique 3D structure regardless of its length. So this way, a single nucleotide change could dramatically affect the strand's electrophoretic mobility through a gel by altering the intra-strand base pairing and its resultant 3D conformation. So this is what we are gonna exploit in this technique. That is, the DNA that contains a sequence mutation has a measurable mobility difference compared to wild-type DNA when subjected to gel electrophoresis in non-denaturing conditions. Now let's see how SSP works. So let's suppose there is a DNA sequence which contains the specific genomic locus in which we are interested. What we are gonna do is we amplify this locus using PCR. So we are gonna get two different products, the wild type and the mutant. The wild type has AT, while the mutant possesses GC. Both types of PCR products are heat denatured. Heat denaturation is followed by rapid cooling and chilling on ice. This leads to the formation of different 3D conformations of single-stranded DNA. The conformations are different because the two single strands are actually facing different degree of intra-strand base pairing, which results in loops and folds, that is giving a unique 3D structure to each single strand regardless of their length. Let's confirm this fact by treating the PCR product mixture with gel electrophoresis. So the gel is gonna separate the two strands of interest. The difference is clear. The single strands of both wild and mutant form actually traveled to different locations in the gel according to their electrophoretic mobility. Let me brief you about the step-by-step guide to single-strand conformation polymorphism. Step 1. Isolation of DNA. Step 2. Designing of primers and perform PCR. Step 3. Preparation of sample for analysis. Step 4. Electrophoresis in non-denaturing conditions. Step 5. Analysis of the data. This was all. Thanks for watching.